Welcome back to Biotechniques on Catalyst University. My name is Kevin Tokoff. Please make sure to like this video and subscribe to my channel for future videos and notifications. In this video, we're going to be discussing the principles of gel electrophoresis, and we're going to be looking at DNA gels, which are tools used to separate molecules of DNA based on their size. Now, hopefully we know that DNA is a double-stranded molecule. We have one strand of nucleotides here and a second strand here. They have complementary bindings. So, for example, this thymine right here has complementary binding to this adenine. That represents one base pair. This right here in red is a cytosine, in blue is a guanine, so that's a second base pair, a third base pair, fourth base pair, and so on and so forth. And as you increase the number of base pairs, you increase the size of the DNA molecule. So, in other words, the more base pairs that a DNA molecule has, the bigger it is. And depending on how big the DNA molecule is, they're going to migrate different distances on a gel. So let's talk about how this works. The way a gel works is you have an apparatus that has a negative charge over here. It's a negative electrode that emits a negative electric field. Down here you have a positive electrode which emits a positive electric field. Now if you notice on the DNA molecule backbones, there are phosphates, and these phosphates are all negatively charged. There's no other functional group or moiety anywhere on a DNA molecule that has any other charge. Everything else is neutral. So overall, because of these phosphates, DNA is very negatively charged. And being negatively charged, if you put it in this electric field where you have a negative electric field up here and a positive one down here, which direction is the DNA molecule going to migrate regardless of its size? Well, it's going to migrate down in this case toward the positive electrode because this negative electrode up here repels the DNA because of negative-negative interactions. And also the DNA molecule is going to be attracted towards the positive electrode because you have attractive forces, the negative DNA molecule from the phosphates and the positive electric field being emitted by the positive electrode. So it's twofold, repulsion from the negative electrode and attraction towards the positive. So all the DNA molecules are going to migrate the same direction. Okay? But how do you actually make the gel? What is it and what does the result look like? First of all, the result usually looks like something over here. This is actually what it will look like under an imager. The DNA gel is made from a substance called agarose. And a lot of times you'll hear a percentage gel. For instance, a very common gel is a 1% agarose gel. Now, the way that you actually make a 1% agarose gel is it's 1 gram of agarose in 100 milliliters of a particular buffer, and the type of buffer you use depends on the lab. The two most common ones are TAE buffer and TBE buffer. The lab that I am in uses TBE buffer, but you actually mix this up, 1 gram of agarose in 100 mils of one of these buffers. Okay. For example, if you had a 2% agarose gel, it'd be 2 grams of agarose in 100 mils of this buffer. Okay, but the most common one is 1%. And then what you'll also have is a small amount of what's called a DNA intercalating agent, or an intercalating DNA dye. And the three most common ones that are used are ethidium bromide, or just called ethidium, gel red, and gel green. The difference between these, ethidium is normally the cheapest, as we'll come back to, and gel red and gel green are a little bit more expensive, but there's trade-offs. These two, the gel red and gel greens, are not carcinogenic, meaning they do not cause cancer. Ethidium is a known carcinogen, cancer-causing agent, so there's trade-offs. It's cheaper, but it causes cancer. Well, we'll come back to these intercalating agents in a minute, but essentially what happens is you mix all this up and you make the gel. The gel will have to solidify because it's initially a liquid. You pour the gel, it solidifies, and once it's solid, you can actually load DNA into each of these what are called wells up here at the top. So initially, you don't have any of this separation, but the DNA will all be located in these wells up at the top. Again, the negative electrodes up here, positive down here. In one of these, normally what you use is what's called a molecular weight ladder or a base pair ladder. This is a standard ladder where each of these bands right here, these are DNA fragments. They each have a known size. So for example, this one down here, this DNA molecule, which is it comes out usually in white on most imagers. This will actually be 100 base pairs. This is 200 base pairs. Up here, maybe this one's 1,000 base pairs. Okay, so the point is this molecular weight ladder, these are known base pair lengths. And so whenever you electrophorese it, you can actually tell the number of base pairs in each of these bands in your samples. And so generally next to the ladder you load all of your samples. For example, sample 1, 2, 3, and 4. Initially everything's in the wells, but once you apply the electric field, in other words you basically turn the machine on, this negative electrode starts emitting a negative electric field, this positive electrode starts emitting a positive electric field, and just as we talked about on the previous slide, the DNA molecules start migrating towards the positive electrode. All right, so that's what you see here. This is the end result. So notice that each of these DNA molecules, which is represented by a white band, they all migrated a different distance, okay? And that distance that they migrate is inversely related to the size of the molecule. In other words, it's inversely related to the number of base pairs. So the DNA molecules or fragments that have a lower number of base pairs that are smaller, they migrate further. Okay, so they're going to be down here. The larger DNA fragments, such as the bigger ones, we mean more base pairs, they're not going to migrate as far. They're going to be up here at the top. And the way to think about this is you can think about it in one of two ways. You can think about it in terms of Newton's second law. So if you apply the same amount of force to all of these, if something has more mass, it's not going to accelerate as quickly, right? So if it has more mass, so more base pairs like this A fragment or even this E fragment, they're not going to migrate as quickly. They're not going to accelerate as quickly, so they're not going to migrate as far. Whereas the smaller ones are going to go much further because they have less mass, so they accelerate more quickly in this electric field. The other way to think about it is just in terms of everyday life. What typically accelerates faster, a small sports car or a big truck? Like a semi-truck. Well, the sports car is going to accelerate quicker because it's smaller. Okay, the semi-truck is huge in comparison, so it's going to accelerate much slower, and so it's not going to go as far. Okay, another way to think about it, if you go up on the track in the gym, and in one case you try to run a lap with a 5-pound dumbbell in your hands, you'll probably be able to move really quickly. If you then go up on the track, run the same distance, but you try to hold a 50-pound dumbbell, you're going to be moving a lot more slowly. Okay, so again, bigger DNA fragments don't move as far. Smaller DNA fragments move much farther. And so that's the principle here. And actually, we'll do a video eventually on protein separations. And again, it'll operate by the exact same principle. Now, even though we can already see these bands here theoretically, before we actually get into how they're visualized, let's actually talk about how we interpret the sizes of each of these bands, A through G, and talk about which one's the smallest and the largest. So the way you figure out the sizes of these is first of all, you will need to construct a standard curve. We'll talk about that in a separate video, but you can at the very least from this estimate the sizes. Okay. So for example, what's the approximate number of base pairs in band A? Well, it's right on level with this standard 10,000 base pairs, so A is probably around 10,000 base pairs. B, again, it's between these two, so in order to get an exact base pair count, we'd actually have to construct a standard curve, which we'll do in a different video, but I can at least say that this is somewhere between 200 and 300 base pairs. C looks like it's between 1,000 and 1,200 base pairs. But D looks like it's right at about 100 base pairs. E and F are right at 2,000 and 1,500 base pairs respectively, and then G is around 500 base pairs. So again, if you have a band like B or C where those are between two standard markers right here, they're between them, you can't say exactly where it is. If you really want to be sure, you actually have to construct a standard curve, and again, watch for that video because we'll have something on that. We can also ask questions like which DNA band is the smallest? Well, in a gel like this, the smallest band is usually going to be the one that goes the furthest on the gel. So it's going to be the furthest away from the wells towards the bottom. So D is going to be the smallest DNA fragment here, or the smallest band, as we would say. If we ask which is the largest DNA molecule, that's going to be the one that migrates the least distance from the wells. In that case, this is going to be A right here. If you ever try to separate entire chromosomes without a PCR amplification, which we talked about in the previous video, entire chromosomes are very large, and so they would be expected to be up here by the largest molecular weight marker, very, very close to the top. All right? So hopefully the answers to these three questions make some sense. All right, now let's talk about how we actually visualize these. How do we actually get these bands right here? And it turns out that it's from emission of ultraviolet light in an imager. But remember, what I mentioned is that earlier, the gels that you make, when you make the gel, somewhere in the gel we have what's called an intercalating DNA dye, such as these three right here. And that's what we're going to talk about on this slide. Here's the molecular structures of them. Over here on the left is a much smaller one called ethidium bromide. This one is a cancer-causing agent. It's very carcinogenic. It happens to be cheaper, so a lot of labs that don't have as much money will probably use ethidium bromide. These two are more expensive. Gel red and gel green. They have slightly different chemical structures, particularly on the aromatic regions on the ends, but these are not known to be cancer-causing agents, and actually they have greater sensitivity than ethidium bromide. So actually, if your lab has enough money to pay for these are actually better dyes, not even just because they're not carcinogenic, they're also more sensitive. And the basic idea of what these dyes do is they intercalate between the DNA strands. So here's one strand right here in my mouse's. Here's the second strand. What you can see here is that the ethidium bromide is actually getting sandwiched between the two strands of the DNA. Gel red and gel green will do something very similar, and when this occurs, there's some electronic interactions, and what will happen is these molecules' fluorescence wavelengths will actually change a little bit. And what you can do is you can actually shine UV light. That's what the imager does. It'll shine UV light on the sample, and what will happen is these molecules, ethidium bromide, gel red, and gel green, will initially absorb that light, and then they will fluoresce, meaning they emit light of a certain wavelength, and you can actually detect that wavelength of light, and ultimately, wherever you see the fluorescence, that means that's where the ethidium bromide is, that's where the gel red is, that's where the gel green is. But that also means that's where the DNA is. So if we go back to this gel image right here, this is where a DNA molecule is because the ethidium or the gel green or whatever we were using intercalated between the DNA strands right here and causes that region of the gel to fluoresce. Okay, there really is gel green or gel red or ethidium depending everywhere in this gel because that's how you made the gel, but it doesn't really have that change in wavelength until it actually gets intercalated between the DNA strands. So that's why you don't see white everywhere here, okay? That's why you don't see bands everywhere because it requires the intercalation between the two strands, and then when you visualize it through the imager, you get something that looks like this. Depending on the imager you're using, they may look a little bit different. In some cases, the bands can actually be a little bit on the purple side. It just depends on the machine, but you should in any case get something that looks like this. Alright, so hopefully at this point you understand the principles of DNA gels and gel electrophoresis. In the next video, what we're actually going to do is construct a standard curve, and what we'll be able to do is actually get the approximate molecular weights, or I should say number of base pairs, for bands that lie between the standard markers. And what you'll find is if you actually do this technique in the lab, it's very rare that these actually line up exactly with a standard marker. Most of the time they're somewhere in between, so if you're really concerned about the number of base pairs, you actually do have to construct a standard curve. Alright, please make sure to like this video and subscribe to the channel for future videos and notifications. Thank you very much.